Hello, everyone. Thank you for joining today's webinar. My name is Dana Haley Vicente, and I'm going to be talking about how to make your own cell-based assays to study beta-arrestin recruitment. Also includes how to make your own cell lines as part of that. The webinar is about 25 minutes. It is being recorded. You'll get the recording and the slides after the talk. Let's see, your phones are muted. If you have any questions, please use the WebEx chat box, and I will get to those questions at the end of the talk. If I don't get to your questions, I have your email address, and I will email you directly with your answers, okay? All right, so let's get started. Today's talk is broken up into three main sections. The first section will go over the basic biology of beta-arrestin. Then we'll follow that up with the tools that you need to use to create your own beta-arrestin cell lines and cell-based assays. Next, I'll go over the general experimental design, followed by workflows and protocols, and then end up with a review of about 4 beta-arrestin recruitment applications focused specifically on GPCRs. So what are beta-arrestins? They are cytosolic proteins, ubiquitously expressed, and elicit multiple functions. There's about 4 known arrestins, visual arrestin, cone arrestins, and then beta-arrestin 1 and 2. Traditionally, most of the papers have been focused on beta-arrestin 2, so that will be the topic of today's discussion. If you focus on the first picture at the top, the beta-arrestin 2, they form complexes with 7 transmembrane receptors, such as GPCRs. And after the ligand stimulates and the GPCR gets phosphorylated by GERK kinases, the arrestin's primary role with GPCRs is to block the G protein signaling and mediate internalization, which includes the desensitization and endocytosis of the GPCR, making it unavailable for further activation, unlike with G protein-dependent signaling pathways. Another role is to couple these same receptors to alternative signaling transduction cascades. If you see that pie chart at the bottom, main role is linking to binding to kinases. There's also nucleic acid binding proteins, phosphatases, even trafficking proteins, cellular organization, metabolic enzymes, and many more. So for a full list, I would like to refer you to the reference at the bottom of the slide. The second one has a nice list there. All right, so why are we discussing, how to create your own beta-arrestin cell-based assays? And there is a definite need for other assays available, knowing that most of the assays today focus on, for instance, GPCRs, and not very many are found for non-GPCR beta-arrestin binding proteins, like I mentioned in the last slide. Alternatively, you may want to study tissue-specific variations, looking at different cell types, not just the typical CHO or HEC293 cells that most assays are based on. Maybe you also want to compare different orthologs, so looking at GPCRs from different species, other than the typical human or even mouse and rat, which are most of the assays out there are focused on. So, and then also maybe you have a GPCR mutant that causes a specific disease and an assay is not available for you. So this is a nice way, or you'll see the tools to create your own cell lines and cell-based assays to study all these needs. All right, so next we're going to get into the tools to create your own cell lines and cell-based assays. But first, to understand the tools, there's a technology behind them that I'd like to go over in just one slide. This technology is called enzyme fragment complementation. It's used to create cell lines and assays to study protein-protein interactions and other applications, and not just specifically for, our particular discussion today for GPCRs. It works on many other applications. EFC is very nice. It allows for stable detection of transient interactions. It is a non-reversible reaction. It's low affinity complementation, allows for natural protein-protein interactions to occur and to drive the reaction. It's been validated, over almost 2 decades now, where there's hundreds of peer-reviewed publications available. It is a highly optimized system, especially for GPCRs. We have validation data for over 90% of the GPCR targets and a handful of non-GPCR 7-transmembrane receptor targets as well. The system, if we focus at the bottom, it's a very easy to use, homogeneous system, endpoint assay that doesn't require control of reaction time. So in the image, you see that the EFC technology is based on a split beta-galactosidase enzyme. These 2 recombinant beta-gal fragments, one is a small peptide fragment, you see as the red squiggly line, and then a larger protein fragment called the enzyme acceptor. So the enzyme donor and the enzyme acceptor. The enzyme donor in particular, we call this a ProLink in this particular application. And this is what you will use to tag your beta-arrestin binding protein to, so your GPCR of interest. The tag does not disturb the protein structure or the cell surface expression or the pharmacology. The enzyme acceptor now in this system is going to be pre-bound to beta-arrestin, and so it will be ready to use, and in the tools that we'll discuss in the next slide. So separately, these beta-gal fragments are inactive, but when they combine, driven by the natural protein-protein interaction, they form an active beta-gal enzyme, which hydrolyzes substrate and produces a nice chemiluminescent signal to have you quantify your interaction and look at the pharmacology of your ligands you may be screening. All right, so first of all, what are the tools you'll need to create your cell lines and assays? There's 3 main tools. The first one is beta-arrestin retroparticles, and these are actually retroviruses expressing beta-arrestin 2 that is fused to the larger beta-galactosidase enzyme acceptor. The little image is at the top. You can infect any dividing mammalian target cells with this. It's a very high efficiency, and as well as this product comes with the confirmation reagents to determine how well the transduction efficiency has went. The next tool is going to be your cloning vectors, and this is that word ProLink, which corresponds to the small tag or the enzyme donor that you will use to do cloning with your GPCR or beta-arrestin binding protein of interest. You'll use this plasmid then to transfect your target cells. And then finally, for the assay part, you're going to need detection reagents. So this is the substrate reagent I mentioned in the last slide. Then you'll provide your ligands to do your screening and obtain your pharmacological data within minutes to several hours. So I'm going to have a slide for each of these tools, and then we'll move right into the protocol. All right, so the retroparticles allow for, the most rapid generation of these cell lines within a few weeks. And it's at a very low cost, so implementation into your lab is very fast. The actual product itself comes with 4 vials of the retroparticles, which are Maloney marine leukemia retroparticles or retroviruses. They're ready to use, ready to infect your cell lines. The transduction efficiency is up to 90%. And again, it contains the beta-arrestin 2 already fused to that EA beta-gal reporter fragment. Also in the kit comes with the confirmation reagents already, and this is for doing the quick validation of the beta-arrestin EA transduction, retroviral and transduction. And then you also get a user manual with easy-to-follow protocols specific to GPCR, beta-arrestin cell line, and assay generation, but it can be easily applied to other beta-arrestin binding proteins. All right, next we have the cloning vectors. So again, ProLink stands for the enzyme donor, the small fragment. These are plasmids for stable transfection of clonally derived cells. There are 4 available. Only 1 plasmid is needed for the creation of your cell-based assays to study, any protein-protein interactions and in our case, beta-arrestin-2, to your GPCR of interest. So we'll use this vector and introduce your target protein of interest, the GPCR, and fuse it with the small donor or prolink tag. The 4 different tags available are listed in the table below, but the first one is by far the most common. It is a low affinity tag. It's called prolink1, and it's for standard protein-protein interactions. We also provide other tags just for weaker protein-protein interactions. This prolink2 one is a higher affinity tag, and then we have 2 variations of the PK2 tag. Uses an ARM sequence is added to it. ARM stands for Arrestin Recruitment Modifying Sequence, or this is a GERK kinase phosphorylation site, which is a specific site for beta-arrestin recruitment binding to help improve the signal-to-background ratios. Ultimately, the guidelines for choosing the correct vector is indicated in the user manual, and we recommend to start with PK1 if your system is new. And then finally, you need the detection reagents to do the cell-based assay and screening of your ligands of interest for beta-arrestin recruitment. These reagents are very sensitive, and they provide quantitative detection of the EFC-based protein-protein interaction. They give ligand-induced chemiluminescent signal that is stable for hours and allows for robust screening and readout on any standard luminometer. The highly validated system, as I mentioned before, with reproducible results. It requires no need for optimization, doesn't require control of the reaction time, and it's really great for QC groups needing lot-to-lot consistency. And it is scalable up to 1536 format and beyond. Overall, this is, if you look at the picture at the bottom, the protocol is a add, mix, and read protocol, so very simple. It's dye-free, non-radioactive, homogeneous, meaning no wash. All right. So let's move into the experimental design and review the protocols and workflow that you may use to create your cell lines and cell-based assays. All right, there's 3 basic steps you see here in the text, 1, 2, and 3, corresponding to the same numbers in the image. So the first step, you simply infect your target cells with the retroparticles, noting that your target parental cell line must be a dividing mammalian target cell line, like CHO, K1, HEC293, A549, et cetera. If you need a list, we can provide that for you. Before the second step, you need to select your PK vector, for instance, PK1, and subclone in your GPCR, just using standard cloning methods. So the second step, you transfect your arrestin EA cells with your plasmid and select for stable GPCR expression. And then finally, you perform the GPCR beta-arrestin recruitment assay with your ligands of interest and obtain your dose-response curves and pharmacological profiles, et cetera, EC50s and S signal of background data. So the next slide, I'm going to go over this in a lot more detail. I'm splitting it up, focusing on steps 1 and 2. Unfortunately, I didn't keep the same numbers. I switched to A and B. So the first main protocol, this corresponds to what you'll see in the user manual. A corresponding to generating a beta-arrestin-2 cell line. Starting with, the cell plating, seed your target cells and incubate overnight. And then that first picture where you infect your target cells with a retrovirus and incubate for about 5 hours. Then there's a recovery stage. You change the media. You incubate for 48 hours. You do an antibiotic selection with fresh media and in the presence of hygromycin. And then you incubate this for about 7 to 10 days under this selection. And then follow this up, highly recommended to use the confirmation reagents to determine if that beta-arrestin EA fusion protein is being expressed. Next, you may want to clone the cell line, freeze it, back up for later, or use this for comparison studies. All right, let's move on to protocol B, where you develop your GPCR beta-arrestin-2 assay. So after cell plating, you're going to do, and your creation of your PK, GPCR plasmid, you picked out the right vector, et cetera. You transfect your cells with this plasmid, using standard transfection methods. Then you perform the antibiotic selection step, change the media, add neomycin in this case. After 5 days of selection with that, you add both the neomycin and the hygromycin. Then incubate for an additional 9 to 12 days under the selection of both of them. And then follow this up with doing the assay, the beta-arrestin recruitment assay, checking the expression of your beta-arrestin 2A and GPCR PK fusion proteins, and generally performing a functional response study in the presence of your agonists or ligands of interest. All right? All right, so let's put this in more of a workflow type of scenario. In general, I wanted to introduce you two terms, the difference between a cell pool and a cell line, because you see both of them in this picture. There is an advantages and disadvantages for both. The advantage for a cell pool is it's very fast. You can get results, within a week or so, and use this for qualitative type of assessment to confirm the intermediate steps in the workflow. It's not desirable, though, for the screening or quantitative studies, and this is because this is a heterogeneous mix of cells. It's potentially unstable and may vary over time. So for cell lines, these are homogeneous cell population, very stable, consistent, and robust. Definitely want to use this for your screening and quantitative studies, but it does take considerably longer to generate cell lines. As you can see in the clonal selection steps in the workflow, it's up to 6 weeks. All right, so looking at the picture, you see some dark blue lines. This corresponds to what I spoke about in the last couple of slides, where you're starting with your cell line, you do transduction, you get your cell pool expressing beta-arrestin 2EA, follow that up with transfection going down to the next line. You get a cell pool with your, after transfection, with the GPCR PK and your beta-arrestin 2EA expressing, and then you confirm it using the assay at the bottom. But what's highly recommended is to generate the other two stable cell lines as well. You could do this the opposite way. You could introduce your plasmid and then do the retroparticle selection. But this may not be preferable. To do it as we've indicated here may be more desirable, because maybe you want to look at multiple comparisons, different GPCRs with different mutations, for instance, looking at different scenarios, basically, in different model systems. So this is the advised pathway to follow. So let's look at this a little bit more in detail. You start out with the cell line, go to the cell pool, and then at this time, once you've done the confirmation, I would recommend moving to the full right and creating your stable cell line. Then moving down to the cell pool, do the confirmation, and then move all the way to the left to create a stable cell line. And this final cell line is definitely used for the quantitative beta-arrestin recruitment assay. All right, so if you have further questions related to any of these topics so far, please enter in the chat window, and I'll try to get them to your questions at the end of the talk. All right, so let's move into looking at 4 different applications for beta-arrestin recruitment. I've listed about 8 of them here. There are additional ones that are on our website if you need that. The first one we'll go over is investigating beta-arrestin recruitment, looking at tissue-specific variations. Knowing that, protein expression is definitely modulated by developmental, environmental, or even pathological type of factors. Pathologies such as metabolic diseases, for instance. So you're going to use your cell line to study these differences in different cell backgrounds. For instance, you want to try it with Jurkat cells or HT29, or instance that represent different tissue types, to evaluate ligand-induced GPCR activation, investigate ligand difference in, definitely unusual cell types that correspond to particular diseases, screen ligands and compare their efficacies, et cetera. So let's look at the example to the right. You're looking at a cholecystokinin A receptor, the GPCR, and investigating the beta-arrestin recruitment in 3 different parental cell lines. The CHO-K1, which is a Chinese hamster, this is an ovary cell line. HEC-293, human embryonic kidney, and then U2OS is human bone epithelial cells. And you see, using the ligand CCK8 is you only see slight differences between these different cell lines, but a slight bias towards CHO-K1 with a 10-fold greater ligand potency, or EC50 in this case. All right, next you may want to analyze mutations. Mutations sometimes block protein-protein interactions or prevent ligand-induced protein activation, ultimately causing diseases or disorders, such as, hyperthyroidism, fertility disorders, different cancers. So you may want to look at differences between the wild-type receptor compared to your mutants, looking at SNPs. Maybe study rare diseases that, due to inherited genetic mutations, identifying ligands that functionally rescue mutant receptors. And that's the one I thought I would use as example today. So looking at the right, you're looking at an adrenergic receptor with a specific mutation, tryptophan to an alanine amino acid, and this causes the specific receptor to get stuck in the ER and not properly traffic to the membrane. So the goal of this experiment was to first rescue the GPCR using a pharmacological chaperone called propranolol. And what this does is it corrects or stabilizes the GPCR mutant, allowing it to properly traffic to the membrane. And these chaperones can be used as drugs as well to correct the system. And then once that GPCR is properly traffics to the membrane, it can be rescued, so you can perform a functional assay using an agonist. In this case, it's procaterol, and that's on the x-axis. Ultimately, the results indicate with the chaperone present, that propranolol, it indicates a response, a nice dose-response curve with increasing levels of agonist present. And what this does, it indicates that that mutant GPCR receptor has been rescued. It reaches the membrane, and it's able to be functionally activated. Without that pharmacological chaperone or pharmacochaperone, propranolol, you see the black curve, there's no response, indicating that it's still stuck in the ER and can't undergo beta-arrestin recruitment in this case. All right, next, we're going to look at, oops, I skipped one. Comparing different ligand responses in different species, so looking at receptor orthologs. One may want to do this to screen ligands for beta-arrestin recruitment, prior to animal studies, looking at human versions of your receptor versus ortholog receptors and other species like non-traditional species, for instance. In this specific example, we're looking at a monkey GPCR, a chemokine receptor versus the human version, looking at beta-arrestin recruitment with the agonist interleukin 8. And you see there is a definite difference. There's about a 10-fold less potency using the monkey receptor compared to the human receptor. All right. And then our final application we'll discuss today is analyzing multiple pathways using the same cell line. This is a huge advantage for all beta-arrestin cell lines. You can perform beta-arrestin recruitment as well as second messenger activation. So you're looking at calcium and cyclic AMP activation type of studies. Maybe you're evaluating ligand biases. You'll run these multiple experiments for instance, on the same plate using the same cell line you generated. This will obviously save you time and money. You can determine which ligand to pursue, as a drug candidate that may have more desirable qualities, such as partial agonists, which may have less side effects. Ultimately, you can obtain pharmacological data, help you to engineer compounds for more desirable profiles, rank your ligands based on potency and compare them to the literature, et cetera. So let's look at two examples of this. The top one is the serotonin receptor. What we've done is created this one cell line and ran a calcium assay, second messenger assay, as well as a beta-arrestin recruitment assay. So you can see the differences, and calcium assays typically have issues showing partial agonists, and only when we ran the beta-arrestin recruitment assay was a specific partial agonist determined. The second example is the opioid receptor, the delta version. We ran a second messenger assay. In this case, it was a cyclic AMP activation assay and the beta-arrestin recruitment assay. And then in the case with the cyclic AMP assays, these typically show saturation effects for some of the ligands. This is just due to the natural amplification system of this particular pathway. So beta-arrestin was allowed, a recruitment assay was nice, and it showed nicely shows the superagonist that wasn't seen in the cyclic AMP assay, as well as a partial agonist as well. Overall, though, both of these assays are complementary to each other. We, recommend you to do both, looking for, different ligand biases, fine-tuning of your ligand pharmacology in general, and the biological pathways. All right, we're doing good on time, so we're going to summarize what we spoke about. We went over how to create your own stable beta-arrestin cell lines and assays. We focused on the tools of a retrovirus-based system that's high transduction efficiency, up to about 90%. It's very rapid to implement into your lab. It's robust. You can get results within 2 weeks. It's highly validated. About over 400 GPCR targets we have to date. There's also a lot of peer-reviewed publications. I have a link here to, you can download this PDF. It goes over the different family members and all the different applications used from target identification all the way to preclinical studies. And then we went over some multiple applications looking at mutants, orthologs, tissue-specific differences as well. All right, thank you for your time, and this recording will be sent to you after the talk. You can always reach me at my email address indicated on this page or through text support if you have further questions. So now I'm going to go to the chat window and look at some of the questions, and I'll get back to you shortly. Hold on. Okay, I got a few questions. The first one is related to the speed of the whole protocol in general, of the workflow. So the viral transduction, takes about 3 days, followed by about 1 to 2 weeks for the selection and then expansion of the stable pool. And then the plasmid transfection takes about 3 days, followed about 2 to 3 weeks longer, so a little bit longer. Overall, going to the clonal cell line at either or both stages will add about 4 to 6 weeks for each cell line derived. The next question is related to why are you using this leukemia, this retrovirus system? First of all, this is the most common viral gene delivery system, this Maloney-Marine leukemia retrovirus. It's a replication incompetent retrovirus that is helper virus free, so it's very safe to use. It's genetically stable for long-term expression, and we definitely obtain high levels of beta-arrestin 2-EA expression in less than 2 weeks from cell plating through the selection step. So very desirable qualities. If you compare this, I got a question related to, why not just a plasmid-based system? Well, we chose to do kind of a combination approach, and we felt that retroviruses, they have up to 90% efficiency compared to plasmid transfections, which is typically around 20%, for instance, doing electroporation. In general, they're more favorable, they're faster, quicker, more efficient, and stable. All right. Okay. For all the additional questions, I will get back to you via email, and thank you again for joining.